Topic of my presentation is a role of molecular communications in growth of glioblastoma multiforme. Molecular communication is a type of communication where information is transferred between biological-based nanomachines through molecules. It has some certain advantages over other type of communications, such as it is feasible, it is appropriate for such a small scale, it is biocompatible, it is also energy efficient, and has a reasonable functional complexity. GBM, glioblastoma multiforme, is defined by the evolution of growing Bionano machine networks with an interplay between the self-renewal and invasion potential of different type of cells, which are glioblastoma stem cells mostly. Our aim is to link the mutual information between various cells to the growth rate of GBM tumor over time, and to investigate whether mutual information can be controlled to control the growth of the GBM tumor. For this purpose, we develop a voxel model where each voxel is representing a single cell in the system, and we consider two different type of configurations named as Go configuration and Grow configuration of cells in the network of cells. As you can see, there are two figures which are series and parallel of voxel model, which define different type of configuration of cells within the extracellular matrix. And what we do is we use this type of model to study the mutual information that is being transmitted from the input cell to the receiver cell. And so overall, the conclusions and future work include that GBM is directly related to the higher mutual information and higher input information flow of signaling molecules. And these results are very high, have a very high clinical relevance, because glioblastoma multiforme has a very high incidence has a very high increasing incidence in the recent years. So future studies will be aimed at designing and development of new therapeutic strategies, theoretically, that will implement models for reducing the tumor growth by controlling the mutual information between the cells. Furthermore, some partners in this project are working on clinical experiments, and this will help us to develop an optimal growth model and for the growth rate of GBM tumor, and which will lead us in the next 2 years to development of molecule-mediated brain drug delivery system for different neurodegenerative diseases, which includes GBM, epilepsy, etc. Thank you very much.